I'm welcoming, I think Janet Nail is here. She's from Phage Africa, and there's somebody else from Phage Africa. So welcome everybody, and I can recognize some of our students as well. And Dr. Hadi from EVA R&D as well.
No problem.
So
we will stick to English. No problem.
It's no problem.
It's always good to make a couple of jokes in Arabic.
Yes, of course. Egyptian phenotype.
Great. And my understanding, Muhammad Hussam, our dear Muhammad Hussam is here as well. You will take care of recording?
Yes, Doctor, I'm with you.
Tamam.
If anyone want to talk, I will allow the mic for him.
Great. He's the guardian of the mic. Now I think as always, especially it's a commute hour, we'll probably give 2-3 minutes after 7, and then I think you're already recording, right? So great. So yeah, we'll just have 2-3 minutes, and then Dr. George, inshallah, will start the introduction.
Sure, yeah, Doctor.
So far, I think most of you know what MARC is, but and who MARC are, but I expect many more people. So yeah, and we have both the chat and the Q&A boxes, right? So they can help us as well. I can just test the Q&A. Well, it's not set up, so we will ask the questions on chat. So we almost have 40 people now. That's a good attendance for the 7 p.m. hour. We are sorry for it's your commute hour and dinner time maybe for some people, or lunch for others.
Even not lunch. Yeah,
and we have Mu'tasim here. I'm glad to see Mu'tasim is here. Yeah, I'm not setting probably the view properly, so I can see some people and the others on other pages. But Mu'tasim is here. He's a science journalist, and he will moderate the Q&A session. And I think Dr. Riad Armanious is here as well. So I think we're good to start.
Hello, everyone.
Hello, Dr. Riad. Yeah, without further ado, I introduce Dr. George Maher, the head of MARC, who will launch the meeting, the webinar.
Okay. Good evening, everyone. My name is George Maher. I'm the head of MARC Research Center. It's my great pleasure to be with you all today and welcome you to our initial webinar that will be a part of our series of designed webinars that help and support to empower the research and the researcher in their journey for discovery. At MARC Research Center, we believe that fostering a collaborative research environment is crucial in advancing science and creating impactful solutions for public health challenges. Tonight, we will mark our beginning of series of conversations where we aim to share knowledge, explore innovative ideas, and build a community that's committed to excellence. Your participation is vital in our mission, and I encourage you all to engage, ask questions, and make sure most of the opportunity to learn is gained. Thank you all for being with us today, and I will be looking forward to meet you all in coming webinars soon, inshallah. And here I will be leaving the floor to our CEO, Dr. Riad Armanious, to share a welcome speech for you. Dr. Riad, the floor is yours, please.
Thank you, George. No speech on my side. it's great to be back to these small discussions, small but very selective, especially on pertinent points. I think the discussion today is one that will, I think, gain a lot of wind with time. First, let me introduce myself. My name is Riad. I'm a pharmacist from Egypt, and I'm the CEO of the group that also has a pharmaceutical company called Pharma. But you know, let's say I was a founder of MARC as a research center, and at MARC we're trying to advance the practice of medicine through the best scientific discoveries and through collaborating with the best scientists. And hopefully this forum and similar fora can help us in doing that. Without saying too much, I know that people didn't come here to hear me. Let me turn it back, and so that we can get on with the scientific content. I'm joining today from Saudi, where recently we've had some good discovery through a collaboration with King Abdulaziz Hospital and University, where polyvalent antibodies were identified and created for monkeypox. This work is under patenting with the US Patent Office, and hopefully we would be able to have such antibodies benefiting a lot of people in the region in a very safe and cost-efficient manner. And such a collaboration is work that took place over a year ago, and has been a very good collaboration that we hope to build on while we produce other antibodies for important targets, both human as well as animal health. But anyways, that's that's a small piece of news from my side. And with that, let me turn it back, and let's please start our scientific main course.
Thank you so much, Dr. Riad and Dr. George. Again, this is a 7 p.m. Webinar, so we don't want to make it too heavy, too formal. However, it's about a pertinent topic, as you heard, and the title of the webinar is Monkeypox and Emerging Health Threats. Are we better prepared? Of course, we were preparing actually our regular webinar series to start in September, and here we go. We have the WHO announces that monkeypox is now a high threat, and we want to hear more. So I'm not going to talk more about MARC after the founder and the head of MARC have spoken, but I think and I'm new to MARC. I am now the head of Bioscience Research Labs. My name is Ramy Aziz, as you can see on my screen. I'm also a professor of microbiology and immunology. The reason I want to just say a word about MARC is that when I was working in research, I received a grant that MARC gave related to cancer cell therapy. So I knew MARC first as a stakeholder, as someone who benefited from this very smart vision from Dr. Riad and the founder, Dr. Samira. And with the vision, and I heard Dr. Riad say it initially, it's that COVID-19, it's one of the main reasons. COVID-19 took us by surprise, took everybody by surprise. But some were surprised and didn't do anything, and others were surprised and started doing things. I don't want to make some analogies, but it's something like a Noah's Ark. Everybody's there is a flood, but some people are prepared. So humanity was really shocked that after all this science, we're not there yet. We're not able to fight a small virus. And Dr. Riad and other visionary people have seen that these countries who can make their vaccine, who can I mean, I don't need to explain this. Like, there is a huge technology gap that we witnessed in COVID-19 in every aspect of health and science and so on. So I think this is how MARC really started, that we need to make our own products as well and not wait in the line for a vaccine or a drug. And this is exactly what we will talk about today. Are we also prepared for monkeypox or any other zoonotic risk that comes here? And I want to say that when when I was younger, when everybody was an undergrad, we could talk about anything. But once we learned some science and get a PhD or a similar degree, we really learned that you cannot speak about any topic. You can actually say, you cannot say something about everything. It's the opposite. You need to really know your specialty. So we immediately looked for who can talk about monkeypox, which is still emerging, and we cannot find better than our two speakers today, Dr. Bassem Toeama and Dr. Ahmed Mustafa. And I will introduce each before his talk, but like, again, this is a very important principle. We really need to seek the experts, because this age is full of self-acclaimed experts who write everything on social media. It's really important to listen to them and see years of experience behind what is happening. But I just want to launch, with one question. Well, I'm going to ask the question in a reverse way. I was going to ask who is vaccinated against smallpox, and then I find it an embarrassing question. It reflects our age. So maybe the opposite. Can you raise your hands if you are not vaccinated against smallpox? You can show some raise of hands. I don't know how. We have like 60 people now, but it's many people are not vaccinated against smallpox, which is good in some sense because the population is young, but it also raises the first question that we give to our speakers while they are going to present: can smallpox vaccine protect old folks like me from monkeypox or not? So without further ado, I will move to Dr. Bassem Toeama. And again, as I said in the informal discussion before starting, Dr. Bassem Toeama was referred to us as a consultant to the WHO. He's an MD, he's a doctor. He has years of experience, almost 25 years of experience in health economics, pharmacovigilance, health technology assessment, and he's a consultant to the WHO, but he also founded his own company. He's the CEO and founder of MD Pharma consulting groups in Canada and in Egypt. And he's currently in Toronto. He's speaking to us from Toronto. But I realized with the name and the resemblance that he is the son of my dear professor, God bless his soul, God have mercy on his soul, Dr. Toeama, a pioneer of microbiology in Egypt, and he was my actually master's advisor, and also Dr. Kamelia, who is our great professor and the former vice dean of faculty of pharmacy, Cairo University. And it's a total coincidence, but we are really welcoming him here. Many of us are students of his parents, and many of us are looking forward to his introduction about monkeypox epidemiology. I'm not managing very much the technical aspects, but I believe that you can share your presentation readily. Otherwise, Mr. Mohammed Hussein will help you with that.
Okay, thank you very much, Dr. Ramy, and thank you all for your attendance. And without further ado, I'm going to start sharing my presentation. So I think all of you can see my screen, right?
Yes, we can see the whole screen.
We can see ourselves. Yes.
Okay.
Now the presentation is clear. Yes.
Okay, 1 second. I'm gonna start from the beginning.
We can do the PowerPoint show mode.
Yes, I'm doing that right now. You can still see me, right?
Yeah, we can see the entire screen
Yes,
with actually the Teams meeting.
Okay, thank you.
But yeah, PowerPoint is starting. Yeah, we can see that as well. No problem.
Okay, thank you.
It's okay. I can understand there is some technical difficulty. So until you said the presentation, I can also introduce the speaker and the moderator, so that we.
Thank you. Until I fix it. Thank you.
Yeah, no problem. So we have also Dr. Ahmed Mustafa Sayed, and Dr. Ahmed is again a dear colleague, a dear friend and professor. He is currently in Texas, which is a country on its own in the US, a staff scientist at Texas Biomedical Research Institute, but he is also an assistant professor of virology at the National Research Center in Egypt, and a pioneer of flu vaccine development at their center. Dr. Ahmed Mustafa Sayed, or we know him as Ahmed Mustafa in publications, he is a pharmacist. He graduated from Faculty of Pharmacy at Zagazig University. And he has a PhD from Germany, from Faculty of Medicine, Justus Liebig University in Germany. And then he did postdoc in several European countries, in Sweden as well. And he received many awards. And the first time I met him, we were sharing an award together. So Dr. Ahmed is also a pioneer of studying COVID in Egypt, and one of the very few developers of an Egyptian vaccine. There were like 2 or 3 versions of them. And we have also Mu'tasim. Mu'tasim is again, he's going to moderate the Q&A session. Mr. Mu'tasim Al-Baroudi, you are here, right? He's one of the talented science journalists in Egypt. He's very interested in science inequity, and he works in the Arab region and in Africa. But he's based in Egypt. He has contributed to different science communication outlets, including Nature Middle East, Nature Africa, and SciDev.net. He's currently very, very concerned with the science and equity gap between what they call the global north and the global south. Dr. Bassem, you ready?
Not yet. I don't know what's wrong with this laptop, so I'm
no
just
problem.
giving...
No problem. Do you mind if we swap? We start with Dr. Ahmed.
No problem at all. It's better. Yes. Thank you.
But we'll ask Dr. Ahmed Mustafa to step in, but not to mention any epidemiological aspect. Dr. Ahmed. Your mic is still muted.
Okay, so now you can see my slides?
Yes, we can see the PowerPoint, not yet the full, yeah, perfect. Yeah, we can see it now.
Okay, so hi everybody. I am honored today to be invited by MARC to present some of our insights about the emerging viruses, include monkeypox and other emerging and re-emerging viral infections in the last few years. So viruses always have these 2 faces, one nice face and one ugly face. So the different viruses that emerged in the last few years, they also showed different case fatality rates, including the bird flu and the smallpox and dengue viruses and MERS coronaviruses and Ebola, and of course the pandemic COVID-19. I will try to be faster for these slides because I prepared a little bit a number of slides, 38 slides. So I would like to be to give some time to Dr. Toeama to present his part 2. So what is the recent emerging and re-emerging viral pathogens that we encountered in the last few years? So the first is highly pathogenic coronaviruses, and all of us know that the Middle East respiratory syndrome coronavirus that emerged in Qatar and then is spread between Gulf countries since 2012. So and this virus has high mortality rate, which is approximately 40%, 35 to 40%. And this virus has emerged in camels. The ancestor of this virus is not known if it is the bats or other horses, but it can transmit from camels to other camels and also to human and lead to, as I told you, the high mortality rate, which is approximately 35%. And also the household transmission between people in the same house, and also to community and health care workers was reported. So in parallel with this, we face the pandemic of SARS coronavirus or COVID-19. And this virus, of course, emerged by late 2019. And since 2019, the virus evolved to many or huge number of variants. So and also the recent one is belong to Omicron is the KB3.11 and KB3 variants, and likely the vaccines which were developed against the KB2 now is not known if it is still effective against the recent variants of the Omicron strain. So the current medications or FDA-approved drugs that can be used to treat the COVID-19 is Paxlovid, which is mixture between two proteases, and remdesivir and molnupiravir. And these are three drugs that are FDA approved. And in parallel, the vaccination platform from Moderna and Pfizer, which include the most recent variant KB2. But this is not the most recent one because the virus is evolving very fast. And now the most recent one is KB3.11. And on the other side, the highly pathogenic avian influenza viruses. So highly pathogenic avian influenza viruses are really has high rate of mortality. So what is avian influenza viruses and what is human influenza viruses? We all know human influenza viruses like H1N1, H3N2 that are circulating every year. And for this, we take the seasonal vaccine. So I will give something like a history overview about these viruses. So most of the human influenza viruses, they emerged, or all of them emerged from avian hosts and transmit to human to lead to several pandemics since 1918 with H1N1, 1957 with H2N2, and 1968 with H3N2, and the Russian flu in 1977 was the re-emergence of H1N1 again, and the most recent pandemic of swine flu in 2009 was H1N1. So all of these viruses were transmitted from avian or swine to human to lead to pandemic. But all of these viruses are mainly replicating in the upper respiratory tract. So so these viruses are converted afterwards after the pandemic to be seasonal influenza viruses, which is less pathogenic, and they are replicating in upper respiratory tract. And for this, these viruses can easily transmit from person to person because with sneezing, they can easily transmit from a person to another. But in parallel, there is some sporadic infections which were caused by avian influenza viruses that are belonging to H5, H7, H9, and H10 subtypes. And the most dangerous subtype is H5 and H7 because some strains of these two strains are is called highly pathogenic strain because they have in their sequence the multi-base cleavage site, so they can easily lead to systemic infection in human. And the risk of these viruses is that they target the lower respiratory tract and lead afterwards to systemic infection, and this leads to high mortality rate, which is approximately 50 to 60% of the cases. This is a video to show that how this infection is working. So I will try to start it.
I don't know if there is supposed to be audio with it. If not, you can comment on it because we cannot hear.
There is no audio? Okay. So the virus, after transmission from person to person, go to the upper respiratory tract, start to recognize the receptor, and then enter into the cells and then replicate. So hijacking all system inside the cells and replicate into millions of variants that can be leads to inflammation of the upper respiratory tract and sometimes the lower respiratory tract if there is something like secondary bacterial infection. And this will enable the virus afterwards also with the high replication in the upper respiratory tract, with the sneezing to spread radially from person to a person to ensure sustained release or sustained transmission between people. And this character is distinct for the human influenza viruses, but is still not established for avian influenza viruses. So we will speak about the history of avian influenza viruses, how they evolved or how they started. So avian influenza virus is the first transmission from migratory birds to domestic and then to human was reported between 1996 to 1997 when outbreak in China happens and some reported cases in Asia in human reported. And then in the second wave, this virus disappeared for some time and then re-emerged again in 2003, 2005 to lead to the second wave of avian influenza virus H5N1. And then the virus undergoes a lot of reassortments to re-emerge again in 2014 and 2016 with a new genetic constellation, which is H5N8 and H5N6. And this virus was the ancestor of the recent or the strain that is widely distributing in Africa, in Europe, in Asia, which is H5N1 of the clade 2344B. So the clades of influenza are too many. So, but this is the most recent clade of H5N1 that is circulating and represent problems in the globally. And if we speak about Egypt, Egypt is considered one of the endemic regions, geographical regions of avian influenza virus because it is one of the busways of the migratory birds, the ancestor of all of these avian influenza viruses. It has around 34 stopover sites all over the Republic for migratory birds. And in these sites, the migratory birds start to exchange the viruses with the terrestrial or the domestic avian hosts. And most recently, which makes the situation more complicated, is that the virus cows was not reported to be host for avian influenza virus or influenza A viruses at all. But most recently in USA, there is many outbreaks that happens with the H5N1 of the clade 2344B. And this virus was originally transmitted. As you see in the slides, this is the genesis of this new virus. It started to reassort between tens of viruses to get this genetic constellation of the genotype B313. And this genotype B313 was very minor reassortant, but this is the reason of the recent cattle H5N1 outbreak in USA. And this represents really high public risk because, as you know, the cow is a source of the milk, the source of meat, the source of different stuff that is used for public, and this leads to a lot of disputes. If this virus can be transmitted through commercial milk, can be also transmitted through commercial meats, and so on. And the situation becomes more worse after, as you see the map of USA, the virus transmitted to many different states in few weeks, and also some states reported human cases. And this is the first human case that was reported in Texas. And the patient showed some conjunctivitis, so he did not develop high respiratory symptoms, but he showed some conjunctivitis, fever, and this is all. But is this virus is low pathogenic compared to the other or well-known H5N1 viruses. We tested this virus in mice, and the CDC tested this virus in ferrets, and surprisingly, the virus was really very lethal at very low infectious doses, up to 10 viral particles, and was also associated with high cytokine storm in the infected or died animals. Yeah. And this represent, that this virus may represent a hazard for if the infection or person-to-person infection is established. To combat influenza viruses, we usually develop influenza vaccines, which are used for the seasonally for the human influenza vaccines. But for avian influenza viruses, the traditional way to generate these vaccines was to use a high growth virus like BR8 virus and the candidate vaccine, which is decided by WHO, infect them together in one egg, and then get hundreds of reassortants. And we use these reassortants or these viruses to test them against sera and find out which virus is expressing the HA and NA of the candidate vaccines. And this process was very time consuming and also costly. But with the emergence or the development of reverse genetic system for influenza viruses, this makes the process more easier. So now what we are doing is we do this process of creating reassortant influenza vaccine strain in lab. In 1 week, you can generate this influenza vaccine strain by including clone segments of the highly replicating virus like BR8 virus. It's H1N1, which is recommended by WHO to be used as a backbone for influenza vaccine, together with cloned HA and NA of the currently circulating or the candidate vaccine strains, transfect them into co-culture of 293 T cells and MDCK cells, and then get the virus to be propagated in embryonated egg or in cell culture. So the accredited cell culture lines for this is Vero cells and MDCK cells. And also this is the propagated virus can be inactivated by beta-propiolactone for human use, or can be inactivated by formalin for avian use, and then formulated with adjuvants to be used for immunization. And on better also, this leads to improvement in the ratio of live attenuated vaccines. So the live attenuated vaccine is the same process, but it's different in the backbone. For the inactivated, we are using the BR8 as a replicating vaccine strain. But for the live attenuated, we use the backbone of temperature sensitive strain like Leningrad strain, H2N2. Together with the plasmids expressing the HA and NA of the recommended vaccine strain, and generates the virus, the 6+2 virus, and also propagate in cell culture or embryonated egg to propagate the strains to be used as live attenuated vaccine. So at the moment, we tested the cattle flu strains and the other avian strains against the oseltamivir, zanamivir, peramivir, and baloxavir, and the recent isolates are still sensitive to all of these neuraminidase inhibitors and BA inhibitors. So these are FDA-approved drugs to be used in case of infection with avian influenza viruses. And in parallel, to be prepared for possible pandemics, or if the virus established already sustained transmission from person to person, CDC is preparing something like stockpile of the vaccine strains. So the current vaccine strains that are used for the recent H5N1, clade 2344B, are influenza, are these two strains. So they have these two strains. Once pandemic situation or large outbreaks started, these strains are ready. They will distribute it between the manufacturer, and then the vaccine can start. The production of the vaccine can be started within few days or few weeks. So, and then arthropox viruses. So the arthropox viruses, Mpox or Monkeypox. This is with the recent outbreak, or so a few weeks ago. The Monkeypox is one of the viruses that re-emerged or emerged and re-emerged in the last 10 years. The host range of this virus is variable, including human and rabbits, pigs, and rats, and also monkeys. So the route of transmission can be transmitted by touching infected tissues or infected subjects, or also what was known that it was highly transmitted between guys in the recent outbreak in 2022. And the incubation period for this virus is variable from 7 to 17 days. I will not speak a lot about this because I think Dr. Toeama will cover this part. So the monkeypox is not a new viral infection. The first reporting of the monkeypox was started in Denmark in 1958. And was reported in monkey used for research and was transmitted from Africa. So the first report in human, it was in 1970 in Congo, Democratic Republic of Congo. And as you all know, that smallpox was declared to be already eradicated in 1980. So the last detection of smallpox was in Somalia in 1977. And then in 1980, WHO reported or declared that this smallpox is not existing anymore and is completely eradicated. And so the monkeypox can be, if we classify it, I will go to this map, the virus evolution. So we can classify the monkeypox strains that emerged in recent years into two clades, clade 1 and the clade 2. So clade 1 is mainly detected in Central Africa, so the Congo Republic. And this was more severe if compared to the strains from clade 2. And we were lucky that the clade 2 outbreak that happens in 2022 and 2023 was related to this clade 2 with less severity. So the ancestor reservoir of monkeypox is not yet known. So it was detected in monkey, but is not the monkey is considered to be incidental host, so it's not the primary host for this. But until now, it's not clear what is the main reservoir for this strain. So the route of transmission, as I said before, that it can be transmitted through contact with contaminated skin, contaminated subjects, or contaminated animals, eating contaminated animals, or having any relations with infected person. So the diagnosis, I think Dr. Toeama will cover this part. During the outbreaks that happens in 2022, the outbreak started in Congo and then also transmitted to different continents and this was more related to monkeypox clade 2, mainly to clade 2. And this is the recent cases that happens in 2024 and also started in different African countries and were mostly related to clade 2 and clade 1, especially clade 1b. Egypt also since 2022, so there is no recent cases that were declared by Ministry of Health, but in 2022 outbreak was one case that was reported. So what is the prophylactic and therapeutic interventions for monkeypox? So most of the people say, okay, the MMRV, which is now used to against measles, mumps, and rubella and varicella, so the chickenpox is protective, maybe protective against. So the reports from CDC declare that the MMRV is not used for smallpox or monkeypox. And the vaccination of monkeypox was depending mainly on some vaccines which are based on vaccinia virus and reported to protect against 85% of monkeypox infection. So before I start working in Texas Biomed, with monkeypox, they obligate me, okay, even if you take any other type of vaccines, you have to take vaccination against monkeypox before handling the virus. So this is highly recommended for people who are working with the virus, for people who did not get any shots against smallpox, and pregnant women. So the two vaccines that now in use, or mainly in use against smallpox and approved also against monkeypox, is ACAM 2000, and this was based on the Dryvax. Dryvax was the first generation of smallpox that was licensed in USA to be used against the smallpox. And after 1980, they kept it. So they fridged it and they kept it until 2007 with the emergence of some outbreaks in Republic of Congo. And then they start to use cell culture-based vaccine of this version of Dryvax, which is called ACAM 2000, and now is in use to treat against monkeypox and also chickenpox and also smallpox. The second type of vaccine, which is the ACAM 2000 is live attenuated, and then there is another vaccine which is GYNNEOS, and this was already non-replicating modified vaccinia virus, which is called MVA vaccine, and can be given to people who cannot take live attenuated vaccine. And this leads to protection. Also, the effectiveness was around 66% to 89%. So what is the challenges to develop vaccines against monkeypox or other viruses? One of the challenges is the cost. So the best animal model for monkeypox now is cyanomacaques. And to do some experiments like vaccine development or testing some drugs in vivo, this is very costly. So it needs a lot of fund, which is maybe not available for people working in research, but is only available in commercial scale. So in parallel to vaccines against monkeypox, there is some antivirals that were also approved to be used against monkeypox, such as tecovirimat, which is used before for smallpox and approved for also monkeypox. And this is a compound that is targeting one of the important surface proteins that are involved in the replication of the monkeypox. And the second treatment option is using the vaccinia immunoglobulin. So vaccinia is one of the vaccinia virus is one of the pox viruses, and it's not known also the origin of this virus. It was already isolated by a researcher and propagated for many passages to be attenuated, and afterwards, so it's not until now is not known what is the origin of this vaccinia virus. So in parallel of the tecovirimat and vaccinia immunoglobulins that can be used during treatment, there is cidofovir and brencidofovir, and these are non-specific DNA inhibitors that disrupt DNA replication of the viruses. So if we are speaking about monkeypox now is one of the viruses that represent high individual risk for the person, but low community risk. So if we would like to classify from risk group 1 to risk group 4, so risk group 1 is a pathogen that represents no or low individual and community risk. Risk group 2 is representing moderate individual risk, but low community risk. Risk group 3 is high individual risk, but low community risk. And risk group 4 is representing high individual and high immunity risk. So if you would like to classify the monkeypox and also avian influenza, which we mentioned before, they are both belonging to risk group 3, which is representing high risk to the individual, but still low risk to the community. So this virus can easily spreading, not as easily as respiratory viruses. The capability to mutate RNA viruses are known to be has more mutation rate compared to DNA viruses, and monkeypox is a DNA virus. So the mutation rate for avian influenza virus is higher compared to the monkeypox, and also availability of effective drugs. So yes, there is some effective drugs and some effective pre-pandemic vaccines, but they are not 100% effective, and this is normal. And both viruses, the avian influenza viruses and also the monkeypox, they show with high host range extension, so they have prolonged host range, infecting human, infecting other also mammalian hosts. And with this, I would like to thank you all for your attention, and if there is any questions or... Hello?
Thank you very much, Dr. Ahmed. Very, very rich presentation, and many of the attendees actually work in either pharmaceutical research or industry, so they definitely took notes, I'm sure, especially those also in our company and in our sister company, Eva Pharma, who are interested in development. For the sake of time, we will keep the questions to the end. Unless there is a burning question, you can post it in the chat, and we will ask it to Dr. Ahmed. I just want to touch back with Dr. Bassem. Is everything good at your end?
Yes, everything is good, and I'm going to share.
So we can switch to your screen.
Thank you.
As you know, we wanted to start with a general picture of monkeypox, but we still have Dr. Ahmed left us some unanswered questions. So Dr. Bassem will talk to us about the public health aspect and the risks of monkeypox and why it's highlighted by the WHO. So Dr. Ahmed, can you stop sharing so that Dr. Bassem can...
Okay, now stopped. Yeah.
Yes, thank you. Dr. Bassem,
can you see my screen right now?
Not yet. Yeah, it's coming. It takes a few seconds. So now we see your full screen. Yes, now we see your PowerPoint. Yeah.
Okay.
And if you can show the presentation mode.
Yes, sure.
Yeah, it's coming up. So again, I remind you, Dr. Bassem is from a medical background. However, he's very interested now in public health, a consultant to the WHO, as well as a founder of MD Pharma Consulting Group. اتفضل يا دكتور until the presentation full mode comes up.
Okay. So yes, so now I think it's showing in a full screen mode.
Yes, it's coming up. It comes up a few seconds later in our end, crossing the Atlantic.
Okay, can you see it, everyone?
Yes, it works now.
Okay, thank you. So thank you all to be a part of this wonderful science webinar. And today's presentation, I'm going to focus on the epidemiological aspect and the public health aspect of Mpox. So Mpox and emerging health threats, are we better prepared? And I thank you for highlighting this, because Mpox is one of the public health emergencies that gained international consideration in the past few months. First of all, a disclaimer. So I just want to mention that I don't have any financial or unethical relationship with the sponsor for this webinar, and it's my pleasure to be a part of this webinar. The agenda for today's meeting, we're going to talk about Mpox. So what's Mpox? We're going to talk about the statistics before and after the outbreak, which started in 2022. Then we're going to focus on the epidemiology of Mpox. So the cost of organism, the modes of transmission, the risk factors, the clinical picture which we might get with the Mpox, the diagnostic workup, and the case definition, and finally prevention and treatment. So what is Mpox? So Mpox, in a nutshell, it's a viral zoonotic infection. So what's a viral zoonotic infection? So this means that there is a host of organism, which is a virus. Zoonotic, so this means it's related to animals, and eventually there is a disease at the end, which is an infection. So this is a simple definition for Mpox. By the way, Mpox is a new nomenclature. So before it was used to be called Monkeypox, and then the ICD-10 terminology changed it to Mpox to avoid confusion, because it's no longer related to monkeys only. So as we will know that the modes of transmission have evolved from being animal to humans, and now it can be, the mode of transmission can involve human to human. So that's why they've changed it to Mpox. The host of organism, though, is still called monkeypox, but I think they will change its nomenclature soon. So the host of organism is monkeypox virus. And as Dr. Ahmed had mentioned, there are two basic strains which cause the Mpox. There is the Central Africa strain, clade 1, subtype 1A and 1B. And Dr. Ahmed gave us a rich presentation about the virus and the recent evolution, mutation, and microevolution that happened to the virus, which increased its virulence. And there is the West Africa strain, the clade 2, subtype 2A and 2B. The Central Africa strain is known to be more virulent than the West Africa strain, and it's more prevalent in the Central Africa, in the Democratic Republic of Congo and the nearby countries. This is the organism. With regards to the animal that's related to the Mpox, so we have two types of animals. We have the reservoir animal. So most of the reservoir animals that harbor the Mpox are rodents. Rodents like what? Like rats, like mice, the Gambian mice and the rats. And there is the host animals. So the host animals, they're mostly monkeys, and hence the name, the monkeypox, the previous name of the monkeypox. Nowadays, a human can be a host because of the human-to-human transmission. So a human can be a host that will transmit the virus to another human being. Also, raw meat or brush meat, as Dr. Ahmed has explained, can be can be in like one of the modes of transmission of the virus. By dealing with the brush meat, the virus can be transmitted to us. The disease is the Mpox, and the incubation period known for the Mpox is usually in the range of 4 to 21 days. So the statistics before the outbreak of the Mpox outbreak started in 2022. Before 2022, we had 3,784 cases and 78 deaths reported in the Democratic Republic of Congo in 2018. And I will focus on the Democratic Republic of Congo because it's the country which has the highest prevalence of the Mpox. In 2018, there were 3,784 cases, whether suspected or confirmed cases, and 78 deaths. In 2019, 5,288 cases and 107 deaths. In 2020, so you can see there is an escalation in the number of cases and in the number of deaths as well. In 2020, 6,216 cases and 222 deaths were reported in the Democratic Republic of Congo in 2020. And in 2021, it dropped back a little bit to 2,841 cases and 76 deaths. Then all of a sudden, in 2022, with the outbreak, the numbers have changed. So with the Mpox outbreak, and now it has reached a situation of being a public health emergency of international concern or international consideration. And the reason for that is that the numbers have evolved dramatically. So you can see that in the time period of January to May 2022, in the Democratic Republic of Congo, there were 1,248 cases that were reported, whether suspected cases and or confirmed cases, and 58 deaths. So only in 5 months we got this huge number of cases and deaths. In 2023, so from January to November 2023, now take care of the number now. We have there was an exponential increase in the number of suspected cases. It reached 12,569 cases that were reported during this time period. And since January 2023 until today, as of today, the Democratic Republic of Congo have reported more than 27,000 suspected cases. Now you can see that there is an escalation in the number of cases since the outbreak has started. Also, the outbreak is not only about the increase in the number of cases, it's also about the increase in the geographic distribution of cases. So Mpox has been reported now in new geographic areas of the Democratic Republic of Congo. And unfortunately in June 2024, now before June 2024, Mpox cases were reported sporadically all over the world. So sporadic cases were noted here and there. So sporadic cases will not change the definition from an outbreak to an epidemic, right? But once you have spread outside a confined geographic location, and once you have moved, the number of cases are increasing in other parts of the world. Now you're worried that the disease will evolve from being an outbreak to an epidemic, and then the epidemic might exponentially escalate and become a pandemic, and we will live the disaster of the COVID-19 pandemic again. Now, the WHO, of course, doesn't want that. In June 2024, there has been 934 cases reported worldwide and 4 cases of deaths reported worldwide. Now, this was an alarm to the WHO, and hence the WHO did not declare an epidemic yet. We did not reach the stage of being an epidemic, but we're not going to wait until the Mpox reaches a stage of an epidemic. Then after that becomes a pandemic. Now we have classified Mpox as a public health emergency of international consideration, yes, a public health emergency of international consideration. We need to focus more on that disease. Now, this is the map which shows the distribution of Mpox cases. I think this map was as of June or maybe July 2024, which shows that the source was the African Center for Disease Control and Prevention. And again, this shows that the Democratic Republic of Congo still has the highest prevalence of Mpox, but it has spread, the Mpox has spread to other parts of Africa, especially West Africa. And until now, and thank God, in other parts of Africa, the Mpox has not spread that much. But again, we're not going to wait until we reach the stage of an epidemic, then a pandemic. So being said, what are the causes that will lead to, and this is like a public health thing, what are the causes that will shift an endemic disease and leads it to be an outbreak, and then evolve the outbreak to an epidemic, and then exponentially escalate the epidemic to become a pandemic. So in order to shift an endemic to an outbreak, you must exceed the outbreak threshold. Then after that, subsequently, the basic reproduction number must increase above 1 in order for the outbreak to evolve to an epidemic, and then this will be followed subsequently with an exponential shift or an exponential change in the epidemic to become a pandemic. So the causes for that, they're the same. So either we get an increase in the virulence of the infectious agent, or we introduce the infectious agent, which is the virus, into a new setting where it has not been there before. Or there has been new enhanced modes of transmission, which were not known before, okay? Or change in the susceptibility of the host response to the infectious agent, or increase in the host exposure to the infectious agent, or introduction of the infectious agent through new portals of entry. Now, the next few slides will talk about the causes that led to the shift of Mpox from being an endemic disease in the Democratic Republic of Congo to changed it or shifted it to be an outbreak. And then now, as we know, it's on the verge of becoming an epidemic, and we don't want to reach that. So the first point is what the virulence of the infectious agent. So this is the infectious agent. This is an electron microscope picture of the monkeypox virus, and as you can see, it has a central dense core with a membrane surrounding it. It's a brick-like, and this is its picture under the electron microscope. The monkeypox virus is the causative organism for the Mpox. It belongs to orthopox virus, which is the genus, which belongs to the subfamily of the Choroidopox virini. And then eventually the big family is the Pox viridae. Okay, Dr. Ahmed talked a lot about the virus and the virology, so I'm not gonna go again and explain this part. The modes of transmission. So now we have a new mode of transmission. Before, the monkeypox used to be animal to human transmission. Now, like when we talked about the causes that will evolve or will shift an endemic disease to an outbreak, there must be a new mode of transmission. And this happened in the last few years. So before, it was animal to human transmission, contact with infected animal body fluids, a bite or scratch from an infected animal. And the animal here is like mostly it's the monkey, the rodents, which are the reservoir for the monkeypox virus, but the host that will be in direct contact with the human and will lead to transmission of the virus to the human. Like there are the monkeys or the primates living in the jungles or in that area of Africa, in the jungles, in the central part of Africa and in West Africa. So either contact with their infected body fluids or a bite or a scratch from their infected animal, and the brush meat, as Dr. Ahmed has explained. Human to human transmission, which is a new mode of transmission. So we see that with close personal direct contact and skin inoculation. Now, this was not the case before, and now we can see it with human-to-human transmission and close personal direct contact and skin inoculation. Indirect contact through the infected fomites, clothing or linens, which have the secretions or the body fluids of the infected human. So the fomites, clothing, or linens who have the body secretions or the body fluids of the infected human, these fomites can be an indirect mode of transmission to another human being. There's the droplet spread. It has been reported that the monkeypox virus can be transmitted through the respiratory tract through droplet spread, and eventually the vertical transmission, so from a pregnant mother to her fetus. Accordingly, the risk factors which we can focus on that may lead to or increase, that will increase, the risk factors will increase the susceptibility of certain humans, and they will be at they will be at a higher risk of acquisition of the virus compared to others. There are community risk factors and there are occupational risk factors. Community risk factors include what? Include because it's, as it has been mentioned and it has been reported, that the Mpox, there was reported cases of Mpox in the gays, bisexuals, and the men who have sex with men. And this is because of the new mode of transmission, which is the direct skin contact and the skin inoculation, direct and close personal contact and skin inoculation. Being an immunocompromised person, like for example HIV patients, this puts you at risk of gaining or acquiring the Mpox much easier than the immunocompetent patient, than the immunocompetent individual. Pregnancy, because as mentioned, like vertical transmission is one of the modes of transmission of monkeypox. Occupational risk factors, so we see that with the research lab personnel working with the monkeypox virus, and the specialized clinical lab personnel who work with the monkeypox virus or do culture for clinical specimens that might have the virus. So these clinical lab personnel are definitely at higher risk than the other people, than other individuals. Clinical picture. So this is a picture of, monkeypox is known for being a rash. It's known for its rash. So this is a picture of a rash where it has reached the stage of scabies, crusting and scabies. So the clinical picture, there are symptoms and signs. Symptoms, there are symptoms for the prodromal stage, which is before the rash appears. And then there is the early clinical stage, symptoms of the early clinical stage, which is shortly after the rash appears. So the symptoms include fever, and we can see that in 85 to 90% of cases, headache in 80% of cases, sore throat, back pain, myalgia in 65 to 85% of cases, fatigue, and lymph node enlargement in 70 to 100% of cases. For the signs, there is the rash, which is the characteristic feature or the characteristic sign of the Mpox, and there is the extracutaneous manifestations. So the rash, it appears 1 to 2 days before, 3 to 4 days after the onset of fever. So it can appear, the rash can appear before the onset of fever, 1 to 2 days before the onset of fever, 3 to 4 days after the onset of fever. Initially, it's 2 to 5. It evolves through different stages. So initially, it's 2 to 5 millimeter diameter macules. Then it progresses to papules, vesicles, then umbilicated pseudopustules. So it has like the vesicles will develop tissue debris or dead cells inside them, and these dead debris will make the vesicles turn into it's not a pustule because there is no pus, and hence the name pseudopustule. These debris will be whitish in color, like pus, but it's not pus. Then the lesions crust over and fall off 7 to 14 days after the onset of rash. The rash appears in the genital, perianal, and the perioral regions. It may also appear in the trunk and the face. Genital, because it's now it's known to be a sexually transmitted disease, so it can appear in the genital region, perianal and perioral, trunk and face, and it's usually painful and itchy. Now for the extracutaneous manifestations, so there's lymph node enlargement as mentioned in 70 to 100% of cases. There are ocular manifestations like conjunctivitis, periocular cellulitis, loss of vision, proctitis or inflammation of the prostate gland, ulcerative pharyngitis, tonsillitis, and neurologic manifestations like encephalitis, encephalomyelitis. If there is a spread of the virus to the bloodstream, so patient might have encephalitis / encephalomyelitis. For the diagnostic workup, so the main purpose of the diagnostic workup is to have a Mpox case definition. So what do we do in order to diagnose a patient with Mpox? The first thing is we check for the rash. Do we have a characteristic rash or not? So any new rash, any of the following, either a macular rash, papular rash, vesicular rash, pustular rash, generalized or localized rash, discrete or confluent rash, which we might get from this rash if the patient gets it from or because of traveling to a country, that might have so a person, an individual who traveled to the Democratic Republic of Congo and then came back to Egypt, for example, and he has this new characteristic rash. So this raises suspicion for getting the Mpox. So if you've traveled to a country that has an outbreak of the Mpox and then you return with the rash, this raises the suspicion for Mpox. Epidemiological criteria. So within 21 days of the illness onset, the patient reports contact with a person having a similar rash, or the patient traveled to a country with confirmed cases of Mpox. So, and this is one of the epidemiological criteria. The lab criteria is detection of the orthopox virus DNA by PCR, or detection of orthopox virus using immunohistochemistry or electron microscope, of course, in the clinical samples. So clinical samples may include body fluids, tissues, body fluids from the patient, from the potential patient. Detection of monkeypox virus DNA by PCR or next generation sequencing. Isolation of the monkeypox virus in a viral culture. And eventually the anti-orthopox virus immunoglobulin M antibody, if it is elevated above like a certain titer or a certain value, within 4 to 56 days after the rash onset. And eventually there is the exclusion criteria, which include either an alternative diagnosis that can fully explain the illness, an individual with some symptoms consistent with the Mpox, but does not develop rash within 5 days of the illness onset, and absence of the lab criteria. So all of that is considered the exclusion criteria. Now, if we want to frame that in a case definition, so we have either a suspected case, a probable case, or a confirmed case. A suspected case of Mpox is a patient who has a new rash, is an Mpox suspected case, is a patient who has a new rash, or one epidemiological criterion. So either he has a new rash or one epidemiological criterion, traveled to a country and then came back, and this country has confirmed cases of Mpox, or the patient was in contact with a confirmed cases of Mpox. A probable case is when you detect orthopox virus DNA or orthopox virus in a culture, or when you detect the anti-orthopox virus immunoglobulin M. This is a probable case. Now, the confirmed case is when you detect the monkeypox virus DNA. This is a confirmed case. So by this, we have the case definitions for the Mpox. When we integrate the previous table for the diagnostic workup, in this framework, we get to know what's a suspected case, what's a probable case, and what's a confirmed case. For the prevention of Mpox, so this is the epidemiological triad, and these are the like what we need to work on to prevent the Mpox. So we need to work on the agent, the pathogen, the virus. We need to work on the host, and we need to work on the environment. So the agent, the interventions that we need to do with the agent is either eradicate the agent, the virus, or genetically modify the agent so as to decrease its virulence. And as mentioned, the monkeypox virus, clade 1 and clade 2, clade 1, 1A, 1B, clade 2, 2A, 2B, there has been 2 studies that confirmed that the virus has mutated and has microevoluted, and hence there is increased virulence of this virus. Now, in order to reverse the outbreak and take it back to the endemic stage, we either eradicate the virus or genetically modify the virus. In addition, we work on the other points which were mentioned: the exposure, the susceptibility of the host, the exposure. The connection between the host and the environment, the connection, the exposure. So we educate the host, the human being, on how to decrease the exposure to the monkeypox virus. We change the activity patterns, and we isolate and quarantine. In addition, we can work on the susceptibility factors for the host. So if a patient is immunocompromised, we take care with immunocompromised susceptible hosts. We isolate the immunocompromised. We isolate the cases, of course, so as the susceptible hosts are not exposed to the confirmed cases or to the susceptible cases. We treat them. So all of that, we work on the different pillars of the epidemiological triad. Now, to take that into action, so the prevention strategies, we work on the virulence of the monkeypox virus. So we genetically modify the virus, or we eradicate the virus, right? And or eradicate the virus. For the settings, so because the virus, when it's introduced into a new setting, this might shift the endemic disease to an outbreak. So in order to decrease the introduction of the virus to a new setting, so we have to do continuous surveillance for that. And once we detect a case, once we have a suspected case, we isolate the suspected case. And once we have a confirmed case, we quarantine the confirmed case, okay? For the transmission, so the modes of transmission, so we engage the community. We talk about the risk communication. We engage the community and we talk about risk communication. And this is in the form of what? This is in the form of like media, in the form of media messages. So in countries where there is an outbreak of the mpox, or it's a public health emergency of international concern, like the mpox, we have to engage the community more than that. We have to give bulletins, and we have to spread the news, talk to the community about the mpox, like the modes of transmission, the how to prevent it, like what do we do, the precautions that we need to take. All of that will be like, this is an overview of how to do it. Of course, getting into details, it will need more time to talk about what are the different methods that we may use for risk communication, to communicate the risk and to engage the community. For the susceptibility, so susceptibility of cases, so the best way to deal with the susceptible patients, like the immunocompromised patients, like the gays, the bisexuals, the men who have sex with men, the pregnant females. So the best way is for pre-exposure prophylaxis is by vaccine, of course, vaccination. But of course, like vaccine for immunocompromised patients, like this is not one of the best modes, because it's and as long as I know, as long as I remember, as long as I know, it's the vaccine which is used, it's a live attenuated vaccine. So it will not be suitable for immunocompromised patients to give the, and probably Dr. Ahmed can have a say on that and can tell us more about that. It's a live attenuated vaccine, so it will not be suitable for pre-exposure prophylaxis. For post-exposure prophylaxis, so again, we can use the vaccine for post-exposure. We can use the immunoglobulins post-exposure, meaning that if an individual was exposed to a suspected case, So for post-exposure prophylaxis, we can give the vaccine. We can give, if the patient is not suitable to receive a vaccine. So for example, if this is an immunocompromised patient or if this is a pregnant female, so better to give immunoglobulins instead of giving the vaccine. And eventually, in countries where neither the vaccine nor the immunoglobulin is available, we can give the antiviral medication. And eventually, for the exposure, to limit the exposure, to work on the exposure, we use the personal protective equipment like the gloves, the mask, the gloves, and this is especially important for research lab personnel and the specialized clinical lab personnel. Wearing the personal protective equipment is very important, also for healthcare professionals, and taking the necessary contact and droplet precautions.
We can, maybe have a couple of minutes.
Yeah, I'm almost done.
Okay.
Almost done. So for the prevention strategy, we have to involve, like for human cases, we have to involve, we do a human case investigation and animal case investigation because there is animal to human transmission as well. So this needs a joint One Health investigation. And if we're talking about globally, at a global level, so this will involve the Food and Agriculture Organization, the World Health Organization, and the Office International what is it called? For the zoonotic disease. There is like a joint office that involves the three organizations: the Food and Agriculture Organization, the World Health Organization, and I don't remember the acronym for the third one. I don't remember its name, but it's responsible for the zoonotic diseases. For the treatment, so the Mpox treatment supportive gear, for that we give acetaminophen and non-steroidal anti-inflammatory drugs. Antivirals, so the best antiviral medication in the market that's being prescribed for Mpox is the tisovirimat, and for special populations there is a special management or special treatment. By special populations we mean pregnant or lactating females, fetus, child or adolescent, and immunocompromised patients, HIV patients. For the indications of the tisovirimat, we give that for the immunocompromised patients with active skin conditions who are expected to flare up with the Mpox, like the psoriasis, like the we may give a tisovirimat for pregnant or lactating females, and for patients under 18 years of age, children and adolescents. And eventually we give tisovirimat for patients with life-threatening Mpox. So those who have Mpox affecting more than 25% of the body surface area, those who have Mpox that have spread to the respiratory airway and led to airway compromise, or led to complications, cardiovascular complications. So those are patients who are considered to have life-threatening Mpox, and these are indicated for the antiviral medication. And by this, I've finished my presentation, so thank you all.
Thank you so much, Dr. Bassem Toeama, and thank you so much, Dr. Ahmed Mustafa. Very, very complimentary and rich pair of presentations. And I know we have a lot of curious people around here. We still have 60 impressive indeed for everybody to hold at post-work hours. Thank you so much all for attending and being here. I know we may have a few questions, and I will leave the floor here to Mu'tasim. Maybe we can take of course we don't want to hold you so long, but we can take 10 minutes of questions. And if anybody has still curious questions after that, we can wait another 5 minutes. but for everybody who have other engagements, so we can have a short Q&A session. Mu'tasim, you're here?
Yes, I'm here. Thank you, Dr. Ramy, and thank you, Dr. Ahmed. And I think you can hear me, right?
Yes, very well.
Okay. Thank you, Dr. Bassem and Dr. Ahmed, for the insightful session and insights. Thank you, Dr. Ramy, for inviting me. I have lots of follow-up questions, but I'll take the question from the attendees first. So I think we have a question from Wafa saying, is there any ongoing research on monoclonal or rodent antibodies for Mpox? I think this is for Dr. Ahmed.
Yeah, Dr. Wafa Tayyib, طبعا, she's one of our professors of microbiology at MIU University. Welcome, Dr. Wafa. Dr. Ahmed, do you have an answer?
Yeah, for this, yeah, there is some research is going, but it's on the laboratory level. So I don't know if there is something more than this going or not. But yeah, so for me, I would like to speak about what I'm doing now. We are mostly trying to find out drug repurposing for Mpox, because this is the easy way to manage the problem of the Mpox now, to look for the FDA-approved drugs, and if there is any drugs that may also affect the monkeypox. So it's this concept of drug repurposing, which that started in was COVID-19, and we continue working on this because, as I told you, we have many limitations to work on monkeypox, especially the in vivo model for this virus. Yeah.
Okay, great. Maybe we'll take another question from Janet. Janet is asking, the uptake of COVID-19 vaccine was very poor in the PAM community. So speaking of Mpox, is there any way to improve uptake by this population? I think the question can go to Dr. Bassem.
The BAME community, I think it's the Black Asian in the US, yes.
Okay, so sorry, can you repeat that again, Mu'tasim, the question?
Yeah, yes, sure. I think Janet is asking about the uptake of COVID-19 was actually low during the pandemic. So here we're coming with the Mpox. Do you think there's any, let's speak of any communities that the uptake is low? How can we improve the uptake generally, and especially in the community? Yeah.
So uptake meaning that the uptake at which level? So meaning that the community was not engaged in taking the vaccine? So,
exactly.
Exactly. So,
yes.
So you're talking about community engagement. So again, so we have to study from what happened with the COVID-19 pandemic. We have to investigate why did the African American community did not engage in taking the COVID-19 vaccine. Okay, and in order to do that, you have to go to the people and ask them. So this needs qualitative research. So you prepare your questionnaires, surveys, and you ask the population, and you ask the community, the people, like what was the major obstacle, or what was your concerns, what was the challenges that you thought of, why didn't you take the vaccine? Like, did you have any specific concerns? Did you think that the adverse events related to the vaccine will be too much for you to handle? So you have to check or to learn lessons from what happened from the COVID-19. And by learning these lessons, you will be able to apply it for future vaccines. So most likely it's related to, so what I understand and what I see from my interaction with the people here in Canada and with the different communities here in Canada, they don't take the vaccines because there's a big group which is known as the anti-vaxxers, and probably Dr. Ramy knows about that. So this big group of anti-vaxxers, they spread rumors and they spread false news about the vaccines. So if the people are not educated, they will hear to the myth that a lot of wrong information will be passed on to the other communities, and these other communities, and because we're not doing our part as research community who wants the best for the people, So we should get involved with the other communities, explain to them, like correct for them the methods that they have, and first know what are the methods that they're exposed to, and correct for them the information. And if you correct for the people the information, they will engage in taking the vaccine for sure. Like, this is common sense.
Great. Maybe I'll have a follow-up question on that. I mean, speaking of Africa, we actually, the issue would be that we don't have vaccines currently. They didn't arrive, and we're expecting the vaccines to arrive maybe next week, according to the Africa CDC. So there has been lots of discussions and lots of people saying that the WHO has actually been late in declaring this outbreak as a global threat, and that have led that the vaccines procurement through Gavi and others to low and middle income countries in Africa have been late. Do you have any comment on that, Dr. Bassem?
So I've shown the statistics. The numbers don't lie, right? So, like, it's not, the WHO is the main concern of the WHO is the global health and well-being of the entire world population. And I represent the WHO, by the way. So I'm telling you what the vision of the WHO is. Like, don't believe anything that's being told to you. And again, like, you have numbers. I showed you the numbers. I showed you what were the statistics before the outbreak and the statistics after the outbreak. And you can see that there is an escalation in the numbers of the suspected and confirmed cases. The number of cases have increased progressively over the past few months. Also, the geographic distribution of the monkeypox virus has evolved in the Democratic Republic of Congo, and it has spread outside the Democratic Republic of Congo, and it has reached other countries nearby in West Africa. So we're not gonna stand like, and hence the name, Are We Better Prepared? And this is the name of the presentation, right? Are we better prepared? Are we gonna stand still, stand still without doing any precautionary action? Or are we gonna do some precautions? Are we gonna communicate the risk to the community? Are we gonna be ready with the preventive medicine? Are we gonna be ready with the vaccines to be given to the special populations, to be given to the immunocompromised patients, to be given to the people at risk or susceptible for the disease, the mpox? It's not that we are overestimating the risk. We're not overestimating the risk, but in the meantime, we're not underestimating the risk.
That's great. I mean, we're just looking for clarifying what the numbers mean. And
yeah, of course. And yeah, may I intervene here just for a second, because well, not everybody was there from the beginning, but I'm re-emphasizing, and maybe Dr. George is here as well, and Dr. Soa, that actually, and this is not totally to advertise anything, but actually the MARC as a research center has been very concerned with monkeypox since 2022, actually, and in addition to other sister companies of biologics connected with the EVA group. So we're also taking what Mu'tasim said, is that as we go back to the very initial question, that if it's like bread, if you don't have your bread, you don't have freedom. If you don't have your vaccine, your drugs, you're not making them. You always wait in the line. So we are working very hard to start breaking this cycle and starting producing, although there are so many difficulties, as you all know, but producing immunoglobulins here, perhaps collaborating with everybody in the region and internationally to get the know-how of making vaccines in Egypt has been a very, very recent activity, but we all believe and hope that it will be common practice maybe by the end of this decade. So again, this is one of the purposes of these webinars, is not only to address the problems, but also to see what can we do as science, both academic and industrial communities.
That's great. Doctor, could you follow up on this? I mean, in terms of challenges for manufacturers in Africa, numbers.
Dr. Ahmed,
yes.
the question is to Dr. Ahmed or to me?
I mean, anyone would like to jump on that would be great.
Maybe Dr. George can talk about this. He's half industry as well.
I mean, I've looked at numbers and actually the capacity, at least for fill and finish in Africa, is more than the demand. But we have an issue of technology transfer, I think. And there's been a recent call from Bavarian Nordic to transfer the technology of the vaccines to African manufacturers. But it seems that according to Africa CDC, only 9 have applied for it, and a few of them have the capacity. And it seems there's lots of challenges. And also an aerial report have said that manufacturers in Africa and whole of Africa don't have numbers of the demand of the continent, for example. That's why they are not encouraged. But I don't have a company, I don't know, so I'd love to hear from you.
Maybe Dr. George can give a quick answer. I mean, none of us, of course, knows exact answer, but maybe
yes, we are working now on the development. We are working now on the development of the mRNA vaccine. Then the production and the commercialization of the vaccine will be coming after that for Africa or for the neighboring countries. But for now, we are working on the development of the vaccine itself. Also, we are having a project for isolation of IgY antibodies for against Mpox. It's another project we are working on it as Eva Pharma. Both projects in phase of development right now, so it's earlier to judge the end results.
Oh, that sounds very promising and great to hear.
Any comments from the laboratory side, from Dr. Ahmed, for example, Dr. Ahmed has the experience of developing vaccines here and there in the US, and I don't want to get into any. I don't want to get into any technical details or bureaucratic, but can you address this as well from your experience? What's wrong? What's missing in the loop of development?
Yeah, I think that the to translate what we are doing on the bench into product, this is very costly, and sometimes also the companies are orienting their targets according to the benefit. So if I did this vaccine or did this drug, how I will get benefit back, because this is even for COVID-19, a lot of companies were hesitating to go into vaccine development or antiviral until some government supported them to go in this part. But without support from on the national level or WHO international level, I think a lot of companies will be hesitating to spend millions of dollars only to help to solve some outbreaks like this.
That's great. Should we take more questions from the comments before we close on?
I would say we can we can take 1 or 2 more questions. I know everybody might be tired, but it's very interesting and engaging. So, and sorry also for our speakers in the US and Canada. I'm sorry as well, because it's your working day. We're taking you from work, but maybe 5 more minutes.
Yeah,
okay,
it's
great.
okay.
Okay, great. I'll be quick. I think Mahmoud Fahim has a question. He says, I'm interested in genetics of the virus and would like to ask about the mutation linked to increased transmission to humans and whether this mutation could affect the effectiveness of the current vaccines. I think that goes to Dr. Ahmed.
Yeah, and maybe we can combine this with a question about the receptor as well. So I can't see the question now, but one of the questions were about the receptor. They go together. What's the receptor?
Okay, so as we said, we have 2 clades for this monkeypox, clade 1 and clade 2, and the outbreak in 2022, as Dr. Bassem also told us, that it was reported that this clade 2, especially clade 2B1, showed some changes or evolution that enables this virus to easily transmit not only to other countries in Africa, but also abroad, to go to Americas, go to some Asian countries. But no one knows if these mutations can affect the efficacy or the effectiveness of the vaccines, because still the effectiveness at its reported effectiveness rates of 85% effectiveness for the applied vaccines. For transmission, it's already known, because this is something we can count from the number of cases in Africa. and outside Africa. And for the receptors, there is no known receptors for the monkeypox, but the internalization of the monkeypox into the host cells can happens through macrobinocytosis or through fusion into the cell membrane. This is known information about the entry of this virus into host cells.
Okay, great. Maybe I'll follow up on that with Dr. Bassem. I mean, Dr. Bassem, you showed us the number that the numbers have multiplied quickly the last few months. So, do we know like what the main factors behind this? Like, is it the virus is evolving? Is it other factors, social factors, economic factors? I don't know. Would you tell us?
So there are whenever there is whenever there is a shift of a disease, an infectious disease, from being an endemic to an outbreak, whenever there is a shift to an outbreak or evolution into an outbreak, and then this outbreak can escalate and become an epidemic and then a pandemic, you have to think of 5 factors. 5 factors. The first one, the virulence of the causative organism. So has your causative organism become more virulent or not? Number 2 is the settings. Has your causative organism been introduced into a new environment, a new setting, new portals of entry into the host or not? This is number 2. Number 3, the susceptibility of the host. Is your host more susceptible now to the organism than before or not? Number 4, the exposure. So has the host been exposed to the causative organism at a higher frequency or at a higher rate compared to before or not? And then number 5 is the I think it's the transmission, the modes of transmission. So has there been any new modes of transmission for the causative organism or not? And if we apply that to the Mpox, you will see that at least 3 out of these 5 criteria, they have changed in the past few years. So you have virulence, so there is a microevolution of the virus, and there is the mutation of the virus, and this has been confirmed in 2 studies, by the way, that showed that the I think it's the Central African strain and the West African strain. I think it's the West African strain. The more virulent one is the Central African strain, which is
the central one. Yeah.
The central one. It's the more virulent one, which is 1A and 1A and 1B. I think that the studies have confirmed that there is evolution and mutation in these viruses, which made them more virulent than before. And that's why the number of cases have increased. The number 2 is the modes of transmission. So now we have a new mode of transmission, which is human-to-human transmission. Mpox did not, or monkeypox before, it was animal-to-human transmission. But nowadays we have the human-to-human transmission. You have the direct personal contact, the skin inoculation, the droplet transmission. So you have new modes of transmission. Number 3 is the I think it's the which has changed compared to before. It's the susceptibility. So nowadays we have, like, if we do the statistics, you will see that immunocompromised diseases which lead to immunocompromised status have increased compared to before. So now we have, like all of you know that HIV is now it's a manageable disease. So unlike before, HIV used to be a lethal disease or a deadly disease. Nowadays, a lot of people have HIV and they live with HIV for a long period of time. Yet still, they're considered immunocompromised patients, and you have to follow up their CD4 count and make sure that they don't drop. The CD4 count for them doesn't drop. The T lymphocytes CD4 count does not drop below 200, because if it drops to below 200, the patients become more susceptible. They become immunocompromised and they become more susceptible to opportunistic infections. So HIV patients, as well as other patients who have other diseases that play with their immune status, like the cancers, malignancies, hematological malignancies, leukemias, lymphomas, you name it. Patients who have immune-mediated diseases, rheumatologic diseases, who take immunosuppressants, like, for example, for lupus or for psoriasis. So glucocorticoids, patients who take steroids, patients who take immunosuppressives that play with the immune system. So this makes those patients at risk of acquiring the monkeypox virus. So you have at least 3 out of the 5 variables. These 3, there has been change in them, and hence the monkeypox virus can easily spread from one patient to another. And with this change in the epidemiological variables, the endemic disease of Mpox has shifted and became an outbreak, and now this outbreak is a public health emergency of international concern.
Thank you very much for the great clarification of that. I think.
Thank you so much. I think, yeah. Thank you, Mu'tasim. Go ahead.
Back to you, Dr. Ramy. Back to you. Thank you.
No, thank you so much. I just wanted to thank you for moderating this. And there are still questions there. Of course, they are in the chat, so the speakers can probably post replies, or later we can. Everything is recorded, and inshallah, the recording, probably it's a little bit of a long recording, so we will split it over 2 maybe YouTube videos that will be released soon. So thank you so much for this, and for staying, like, for the 50 people who are here, staying beyond 8:30. Thank you so much, and thanks to our speakers who gave us their prime time at the midday in the US and in Canada. Thanks, Dr. George, for hosting this first seminar, and for everybody. I see a lot of people from inside and outside the research center, so that's great engagement. Thank you so much, and thanks to Mu'tasim for moderating. Again, we always hope we don't talk about emergencies of health, but maybe inshallah next seminars will be about different topics, so stay on our contact list. One final thank you to all speakers. Please join me using all the emoticons you like, and
thanks a lot, Dr. Bassem and Dr. Ahmed, and thanks, Mu'tasim. Thank you all. Thank you.
Thank you very much. Thank you.
Our pleasure.
Thank you. Bye-bye.
Goodbye.
All right. Yeah, thank you so much. We will end the conversation in a few seconds. Thank you.
Thank you.